talk. So my research area includes bone metabolism, and another is cancer biology and bone metabolism. Today I'm going to talk on phytotherapy for bone fractures. And as I told, we have a plant phytomedicine, which is known to have osteoprotective mechanism. And in this presentation, I'll see the molecular mechanism, how this compound or this extract elicits fracture healing. Well, with the advancement of the science and technology and healthcare, there is a stupendous increase in aging population. As a result, there is a increase in geriatrics related diseases such as dementia, cardiovascular diseases, and bone diseases. Sorry, any question? Are you able to hear me?
Okay. It's okay. We are hearing you. I think,
okay.
Yeah,
okay.
I have muted this. So go ahead, please.
So osteoporosis is one of the main metabolic bone disorder that is associated with a skeletal fracture. And the fracture due to osteoporosis is enormous healthcare burden because millions of dollars are being invested to fix these metabolic bone disorders. Well, coming to the bone is not a dead tissue as it looks like. Rather, it is a very active and metabolic tissue. And it undergoes a process called bone remodeling. Bone remodeling is the process where the bone formation and bone resorption. Resorption is nothing but erosion of the bone. So both are normal physiological process. These both processes are coupled together. If there is any uncoupling because of any factor, that can leads to metabolic bone disorders, including osteoporosis. So the bone remodeling is regulated by various bone cells, and there are primarily 4 bone cells: osteoblast, they're bone forming cells; osteoclast are bone resorbing cells; the bone lining cells; and osteocytes, they are senile or retained osteoblast cells that are after secretion of extracellular matrix, they are impregnated inside the the extracellular matrix actually. So among these, the bone forming cells and bone resorbing cells, they are very important. Osteoclasts, they derive from hematopoietic lineage, whereas osteoblasts, they derive from the mesenchymal stem lineage. Various factors regulates the bone metabolism, which includes numerous hormones, systemic hormones, and various local growth factors. Among these, calcitropic hormones are bone regulating hormones. Parathyroid hormone, calcitonin, and vitamin D3 are very important. And sex steroids, you can appreciate the importance of these sex steroids because during menopause, there is cessation of ovarian function. Steroids, thyroid hormones, glucocorticoids, they also are very much essential for bone metabolism. And various factors like insulin growth factor and Wnt signaling components, they play a very important role in maintenance of bone health. So among the various cell signaling mechanisms, Wnt beta-catenin signaling pathway is very important because they play a critical role in embryogenesis and to the maintenance of adult tissue homeostasis because they regulate cell proliferation, survival, differentiation, migration, and self-renewal of the stem cells. So this schematic diagram shows the Wnt signaling pathway. In the absence of the Wnt ligand or in the presence of inhibitors, the beta-catenin, which is the effector molecule of the Wnt pathway, is phosphorylated by glycogen synthase kinase 3. As a result, it undergoes ubiquitin-mediated proteasomal degradation. As a result, the Wnt pathway is switched off. However, in the presence of the Wnt or in the absence of the Wnt inhibitors, the Wnt binds with Fris receptor and LRP receptors. As a result, the dish protein is activated that inhibits the GSK3. As a result, the phosphorylation of the beta-catenin is prevented and results in accumulation of the beta-catenin in cytoplasm that translocates in the nucleus, where it binds with the transcription factors, T cell factor or lymphoid initiates gene expression. So as I told you, osteoporosis is a metabolic bone disorder that is characterized by microarchitectural deterioration of the bone, resulting in increased bone fragility and susceptibility. So this is the normal bone. You see, the trabecula in the bone. When you see the bone, you can see trabecula. This is normal. This is nothing. It's a normal bone. Where you say trabecular, there are holes in the bone. This is the osteoporotic bone. If the integrity is messed up, it results in porous bone. So osteoporosis affects almost more 200 million women worldwide, and importantly, 1 in 3 women over the age of 50 years, they experience osteoporotic fractures. And importantly, osteoporosis also affects men, so which is really underrated actually. So 1 in 5 men over age, they have the chance of getting osteoporotic fractures. So as I said, osteoporosis is associated with higher risk of fractures, and they cause significant pain and disability. And more importantly, this is associated with severe morbidity and mortality of the patients. Whenever the bone is fractured, it undergoes a normal process of bone healing. It's where the various processes like hematoma formation, soft callus formation, and hard callus formations take place. So the bone healing process is a complex, but it's very well organized, process. There is events of molecular and cellular processes. Who are all the risk for fractures? The people with malnutrition, the poor vitamin D3 or poor calcium, or people who are elderly patients, and the postmenopausal women. That's the main problem. Anyhow, women is going to undergo menopause. Naturally, in the ovarian function ceases. As the result, estradiol goes down, and as a result, they are prone to get osteoporosis. There are people who are independent of postmenopausal, people who take glucocorticoids or any other medicine that can induce osteoporosis, or even idiopathic osteoporosis, they are high risk of getting fractures. Various drugs are used for this osteoporosis as well as fractures. Some include, to mention some, they are bisphosphonates, alendronate or arsenronate, parathyroid hormone replacement therapy, which primarily includes estrogens and selective estrogen receptor modulators. However, these are associated with various side effects in postmenopausal women, which includes hypercalcemia, osteonecrosis, increased risk of cancer, osteosarcoma or breast cancer, gastritis, esophageal irritation, stroke, coronary artery venous thromboembolism. Recently, U.S. Food and Drug Administration (FDA) approved romosozumab, which is an antibody for sclerostin. Sclerostin is a inhibitor of Wnt pathway, Wnt pathway, and studies have shown this significantly inhibits the bone formation. So they have developed, the brand name is Emanity, and this is a humanized monoclonal antibody produced by recombinant DNA technology. However, this is very effective treatment. Naturally, it has high risk of heart attack, stroke, and cardiovascular death. And more common side effects includes joint pain and headache. Always, whenever you use chemicals or the modern medicine, it always is associated with various side effects. And as you all know, it's the father of the medicine, Hippocrates, said, "Let food be the medicine and medicine be the food." Even today, the moderator mentioned about this, and even yesterday's special talk, Dr. Bharat Agarwal also mentioned this. This shows the significance. Hippocrates mentioned this almost 2,400 years ago. It has been appreciated like, the food can be the medicine, and foods primarily from plant sources. If they can act as a potential therapeutic agents. So the phytomedicine is very, very interesting and very important. So currently available drugs, they have associated, it may be either stimulate bone formation or it's going to bone resorption. They all are associated with severe side effects. So here comes the phytomedicine, which has been receiving greater attention in the last few years due to its safe use in the treatment of various diseases, including bone disorders. So various medicinal plants have been used for treatment of fractures in traditional system of medicine. And these are some of the medicines that are used for fracture healing. And mention some Moringa oleifera. It's very common. Many of you may be aware of drumstick leaves. Cissus quadrangulis, they're very commonly used for fracture healing. We have chosen a herb. It's called Armocarpum cochin chinensis, which belongs to the family Fabaceae. It's a small herb found in some forest actually. So these herbs, Armocarpum cochinensis, is rich in various phytochemicals. There are only few studies available where they found they are rich in phytochemicals such as flavonoids, alkaloids, steroids, terpenoids, glycosides, tannins, saponins, coumarins, and they are used in Indian system of medicine to treat bone diseases. However, there are no studies to characterize the mechanism of action of these plant extracts. So we have hypothesized that there is Pharmacorpon Cochin Chinese. Here I'm going to call it as OC. So OC leaf extract may facilitate bone fracture healing by enhancing the bone formation and suppressing the bone resorption by regulating the key molecules involved in bone remodeling. To check the hypothesis, we made these objectives to study the effect of OC on bone turnover markers and to see the expression of critical molecules that regulate bone formation and regulate bone resorption, and also to see the osteoblastic differentiation of bone marrow stromal cells. So we collected the leaves of this plant from the forest from Chengalpattu, Tamil Nadu. It was identified and authenticated by Botanical Survey of India. And we took for initially 250 grams of the shade-dried leaves, crushed into powder and distillated using distillator apparatus, and about 30 grams of extract or something. We used adult female Vista rats, albino rats, 150 to 200 grams, and we induced fracture in that, and the rats were maintained as per institutional animal ethical control. So this is how we induced fracture. Unilateral fracture was induced in the femur by anesthetizing the rats using the scaled-down version of the apparatus. I will show in the next slide. It was described by Bonner and Zahn in 1984. So mid-diaphysis of the pinned femur was fractured by a force generated by dropping, a 500 gram weight from height of 35 centimeter. So the rats were recovered from anesthesia and they were permitted for full unrestricted, full weight-bearing activity. Radiographs were taken to confirm the fractures. So rats anesthetized, cleaning and shaving the femur area. So we made a lateral incision and 1 mm hole is made in the condyles and pin was inserted. The purpose of inserting the pin is otherwise the broken bone will go in a different orientation. You have to keep it in a particular orientation. So that's why we insert a pin, and after the surgery, the radiograph was taken, and this shows the pin properly inserted inside the femur. And this is the apparatus where we draw 5 times of weight to induce unilateral fracture. And we confirm the fracture induction by taking X-ray. So this is where fracture is induced. And we treated these rats with this plant extract. This is the callus formed. Callus is the structure formed at the site of bone fracture. So we used 2 groups of animals, the control and OC treated groups. So the control received olive oil as a vehicle. Initially, we tried different concentration of the OC leaf extract, 200 mg, 400, 800, 1600 mg, but we found like 200 mg induces more bone formation. That's why in this study we have focused only 200 mg concentration. So we treated the rats with this OC extract for 21 days, and we sacrificed it after 21 days, and samples were collected, and the serum samples were used for bone turnover markers. So we dissected out the callus and we use this for RNA and the protein extraction, and we studied for critical molecules that play an important role in bone formation in the callus actually. Coming to the results, so we didn't observe any change in the body weight between the control and OC treatment, not only with this 200 mg per kg body weight, but also in other concentration we tried actually. And we also looked for a microscopic observation of the various organs. We didn't see any abnormality. Shows the safety of the plant extract. So this is the femur bone, and we see very big callus formation in case of OC treated. Most of the the femur we found a good callus formation. We look for the calcium and phosphorus. So hypercalcemia is reflects the bone resorption, and treatment with the OC significantly decreased the calcium level. However, we couldn't find any change in the phosphorus. We looked for the bone formation marker. Alkaline phosphatase is one of the bone formation marker. We found increase in alkaline phosphatase, which is the bone formation marker. At the same time, there was a dramatic, statistically very big difference in the TRAP activity. TRAP is started with an acid phosphatase, which is a marker of osteoclast. So it clearly shows this leaf extract is suppressing the bone for resorption. So on one hand, it is increasing the bone formation. On the other hand, it is suppressing the bone resorption. So then we look for the key molecules that play an important role in osteogenesis actually. So osteocalcin is a calcium binding protein, and RUNX2 is an important transcription factor. It's also called CBFA1, core binding factor 1. So both these, key molecules are upregulated by this OC leaf treatment. So clearly shows it is inducing the osteogenic potential of these bone cells. We look for the 6 steroid receptors. So both estrogen receptor and estrogen receptor alpha and beta play an important role in bone health. Actually, this stimulates bone cell proliferation, differentiation, as well as mineralization. And similarly, androgen receptors play an important role in maintaining the bone mineral density. So interestingly, our OC extract increase estrogen as well as androgen receptor expression. So the possibility may be because of some phytoestrogens present in this extract would have increased the expression of these 6 steroid receptors. We look for the key gene like a bone morphogenetic protein 14, which is also called growth differentiation factor 5. And interestingly, the mRNA expression of this BMP14 is increased by this OC extract. As I mentioned earlier, Wnt signaling is very important, in bone health actually. And I mentioned like the drugs they developed to prevent, the monoclonal antibodies to prevent sclerostin appear to be very effective, though it has side effects. It's effective in preventing osteoporosis and fracture because it suppresses the antagonist of the Wnt. Actually, see here, OC treatment increase the Wnt ligand, which is the stimulator of the pathway, Wnt the beta-catenin, which is the effector molecule. This both ligand and effector molecule are significantly increased by OC leaf treatment. Interestingly, the Wnt inhibitor factor 1, it's totally suppressed by this leaf extract. So it appears to be very promising drug in regulating Wnt pathway and bone. Then we look for the key stemness markers OCT3 and 4, and we look for SOX2 and SOX9. And though we couldn't find much difference in SOX2, OCT3 and 4 and SOX2, the expression was significantly increased by this OC treatment. So clearly shows it also induces the pluripotency of the bone cells. So finally, we checked for the RUNX2 expression. Earlier, we checked this RUNX2 expression in the callus, that is in the bone tissue actually. We isolated the bone marrow from those, femur bone, and we isolated RNA from that, and we checked for the RUNX2 for mRNA expression. In addition to increasing the RUNX2 expression in the bone as such, OC treatment increased the RUNX2 mRNA expression in the bone marrow, which is the source for the stem cells. So this is very interesting. So to summarize, the OC leaf treatment increased the osteoblastic activity as observed by increased alkaline phosphatase, which suggests this OC extract is increasing bone formation. At the same time, it suppresses the osteoclastic activity, the bone resolving cells, as observed by increased trap activity. So the molecular mechanism appears to be increasing the RUNX2, which binds with osteoblast-specific element 2 and triggers expression of various genes that are involved in osteoblastic differentiation. So importantly, the OC extract increase the expression of Wnt3A beta-catenin and also increase the 6 steroid receptor expression, which play an important role in cell proliferation. They also increase osteocalcin and RUNX2 expression, BMP and SOX9 and OCT3 and 4, which plays an important role in differentiation of the bone cells. So, to take it together, our study demonstrated that Ormocarpum cochinchinalis leaf extracts has a potent osteoprotective as well as anti-resorptive activities by modulating the expression of key and crucial factors that are involved in bone remodeling and by activating the Wnt beta-catenin signaling pathway. And this may be the possible mechanism by which OC elicits its bone fracture healing property. So I would like to acknowledge our collaborators, Professor Srinivasan, Dr. Lourdes Camilo, Professor Michael Arundas, Professor Balasubramanian, Dr. Ravi Shankar, the head of the Department of Endocrinology, Dr. Ilangovan, Dr. Shivakumar, and Dr. S. Yuvatach. I also thank the students who worked in the projects, Gautam, Manju, Senthil Kumar, Balaji, and Satish. I thank the funding agency DST for Phase 2, UGC SAP DSA1, and SERP. Thank you for all your attention. I'll be happy to answer if there are any questions.